welcome back to Learning with Isha. We're talking about plant growth hormones. We know what plant growth hormones are. We have already looked at two of them. We have looked at auxins, we have looked at gibberellins, both of which are plant growth promoters. Today we move on to look at another important plant growth promoter, that is cytokinins. Like always, we are going to divide this video in two parts. In the first part, we are going to talk about the discovery. In the second part, we are going to look at the functions. So let's quickly begin. The discovery of cytokinins was a need-based discovery. So let's go back to where it happened. It happened in Wisconsin, USA in the year 1955, where Skoog, Miller and their co-workers were trying to do one thing. Well, what were they trying to do? They took a tobacco plant, okay, and from this tobacco plant, they took out a cell from the pith of the tobacco plant. So they took out this cell, let's say, from the pith of the tobacco plant. Now using this cell, they were trying to perform the technique of tissue culture. We know what tissue culture is. You can take any cell from the plant and put it in a nutrient-rich medium. The medium should have carbon source, nitrogen source, and some plant growth hormones or some plant growth regulators. Exactly, they did that. But yet they could not get a callus out of it. मतलब ये cell divide करके एक ball of cells नहीं बना रहा था. This ball of cells is known as callus. This we already know. So this callus is what eventually differentiates and gives me the whole plant. अगर callus ही नहीं बनेगा तो plant ही नहीं बनेगा. ये callus क्यों नहीं बन रहा था? Because in the medium they added all the nutrients, vitamins, they added auxin, but this medium still lacked a compound. Let's name it as X, and that is the experiment that they were performing. They were trying to find कि इस मीडियम में वो क्या ऐड करे, सप्लीमेंट करे कि ये callus formation हो जाए. So what Miller did was he added a lot of compounds. Finally what he did, उसके लैब में एक बॉटल पड़ी थी of an old DNA. He just took it and he added that in the medium. And to his surprise, it worked. So definitely this old bottle of DNA had something that was causing the cell to rapidly divide. It was discovered that this compound X was actually the degraded DNA having 6-furfuryl aminopurine. So the degraded DNA had a compound known as 6-furfuryl aminopurine that was causing this rapid cell division. On further performing experiments, they found that similar results were also produced when instead of degraded DNA, they either added extract of vascular tissue. Vascular tissue is your xylem and phloem. Also, if they added yeast extract or they added coconut milk. Each of these compounds contained this chemical X that was able to induce a very, very rapid cell division. These group of compounds that were causing a rapid cell division came to be known as kinetins. They came to be known as kinetins. Then how did we get to the word cytokinins? Well, the word kinin also exists in biology. So to avoid confusion with the word kinin, the word cytokinins was given. Now, are these compounds kinetins actually found in the plant? They are not. So what is found in the plant is our next topic. Even though kinetin was not found in plants, the search for a kinetin-like molecule in plants was continued for a long time. Finally, it was found by Latham that the endosperm of the corn में जो milky part होता है ना corn kernel में, that is known as the endosperm of the corn, it contained a kinetin-like molecule This kinetin-like molecule was actually called zeatin by him. Zea comes from zea maize, that is the scientific name for corn. So zeatin is actually the first cytokinin that was discovered from plants. Now let's move to the second part of the video where we'll start talking about the functions of cytokinin. As we have already seen in the discovery experiments that the first most important function of cytokinins definitely has to be that it helps in rapid cell division. So where actually would cytokinin be found in the plant? As you would expect, in those areas where the cells divide very rapidly. We know in the root apex and the shoot apex, we have very fast dividing cells known as meristematic cells. So definitely a lot of cytokinins are going to be found in the root apices and in the shoot buds. As in if this is your plant, this is going to be your shoot apex and this is going to be the tip of the root known as the root apex. You'd find a lot of cytokinins here. Also in the young fruits that are undergoing development, there would be a very large number of cell division. You would also find a large amount of cytokinins in developing or in the young fruits. Besides that, the second important function is that cytokinins help in the lateral shoot growth. What do we mean by lateral growth? See, as much as it is important for the plant to increase in height, it is also important for the plant to gain some width. So the gain of height actually we've seen is done by auxins and gibberellins, mainly by performing enlargement of the cells. So the cells elongate in size and therefore the plant increases in length. But if the plant has to increase in width, it is not going to be by cell elongation, it is going to be by rapid cell division. And we know that rapid cell division is the function of cytokinins. So, the cytokinins are going to help the plant gain width by cell division rather than cell elongation. So, these are known as the actual or the true cell division factor. Another important function of cytokinins is that it helps overcome the phenomenon of apical dominance. Apical dominance, as we've seen, is the phenomena where the presence of this apical bud, the bud present at the shoot apex, what it is going to do, it is going to promote the plant to only gain length. The plant is not going to broaden, it is not going to have more lateral branches. So the lateral buds are not going to develop in the presence of this apical bud. Cytokinins are going to help overcome this phenomena. So इसका क्या मतलब है कि अगर आपका apical bud present है और आपने cytokinins डाले, so that is going to still promote the formation of the lateral branches. This is beneficial in plants like tea, where we require the plant to have a larger number of leaves. The next important function is that it delays the senescence. What is senescence? Senescence is basically the process where any mature leaf or the mature part of the plant eventually is shed off from the plant. It falls off. We know that the old leaves fall off. So what is the process? If we know that a leaf is to fall off, plant first of all will cut off nutrients to that leaf. उसे पता है उस leaf ने गिरना है, उसे और nutrients क्यों देगा वो? So number one, the nutrients will be cut. Number two, whatever chlorophyll is there in that leaf, it is going to be broken down. And chlorophyll से जो भी reabsorb हो सकता होगा, the plant is going to reabsorb that and use that for forming the newer leaves. So eventually chlorophyll breakdown will cause yellowing of leaves and these yellow leaves fall off. What cytokinins do is that they delay the falling of leaves. How will they do it? Well, they increase the nutrient mobilization in the plant. So what is this nutrient mobilization? Mobilization means movement. So they are going to increase the movement of nutrients in the plant. They are going to increase, let's say, the translocation in the phloem. So the phloem now is going to carry more nitrogen, more food. So more food is going to get available to the leaf. Therefore, it is going to stay on the plant for longer. So whenever we want the leaves of the plant to, let's say, greener for a long period of time, we're going to spray cytokinins that are going to help delay this senescence process. Another important function is that cytokinins promote the formation of new chloroplasts and also new leaves. So, well, this was about cytokinins. In the next video, we're going to look at another group of plant hormones that do not have promoting activities. Instead, they have inhibiting activities. So, in this, we're going to cover abscisic acid and ethylene. Thanks for watching the video. Do consider subscribing to the channel. And don't forget to press the like button. Thank you.